मैं Rajveer Singh Sheoran, Associate Professor in Chemistry, SRR Morarka Government PG College. First of all, good afternoon to all the students of MSc Final. Today, we will start the fourth unit, Biophysical Chemistry. So, in this, we will discuss today biological cell and its constituents. First of all, we discuss what is biological cell. Since ancient times, persons like Aristotle too realized that all animals and plants, however complicated, are made of a few elements which are repeated in each of them, though they are referring to organ systems. Later studies showed that organisms are made to even smaller repeating units, the cells. All living organisms, small or large, simple or complex, primitive or advanced, are made up of cells. Thus, this in itself is an example of uniformity among such a large number of diverse forms. The cell also brings structural and functional stability to these organisms. So we can say these cells described as a unit of biological activity delimited by a semi-permeable membrane and capable of self-reproduction in a medium free of their living organism. The cell is the basic structural and functional unit of all living organism. Each cell in itself is self-sufficient in that it has its own structure, metabolism and reproduction. So, unicellular organisms made of a single cell are no less than an independent living body. Very recent studies have revealed the use of an electron microscope and different centrifugation to find the presence of various structural units of the cells with a view to have an idea of their function too. The structure of a biological cell is shown in this figure first. So, we discuss the cell constituents, means first cell membrane. The boundary of every cell has a thin membrane of thickness 75 angstrom units, although the thickness varies from 70 to 100 angstrom. This is also known as unit membrane or plasma membrane, which is formed by the ingredients of the plasma cytoplasm. The membrane consists of three layers: inner layer 20 angstrom of proteins, middle layer 35 angstrom consisting of phospholipids, cholesterol, and the outer layer 20 angstrom consisting of proteins and polysaccharides. These are arranged continuously to form pores of about 8 angstrom. The phospholipid molecules in the light band are arranged in two rows such that the phosphate containing ends, the polar or hydrophobic ends points to the outside, while the non-polar or hydrophobic ends points to the inside. The orientation and nature of the lipid protein component in the cell membrane determines the charge in the membrane and also studies the moments of molecules across the membrane. The surface of the cell membrane bears pores of internal diameter 3 angstrom and length about 75 angstrom, which leads from cytoplasm to the exterior. As seen in the structure of biological cell, there is centrally the nucleus and then pores, mitochondria and nucleus, ribosomes as shown in the figure first. So, the most important function of biological membrane is to restrict the exchange of substance among various compartments, so different types of body fluids are separated from each other by the different membrane. This is done by selective permeability of the various membranes, for which certain substances are passed through different rates, that is high, moderate, or low, while others are prevented from passing through. And the nucleus, secondly, we can discuss the nucleus is the heaviest component of the cell and is separated from the cytoplasm by a double membrane. The outer membrane is continuous with a endoplasmic reticulum. The two membranes are separated from each other by perpendicular 150 angstrom and joins the intervals to form pores which allows the passage of material from the cytoplasm to the nucleus and vice versa. The pores on the nucleus membranes are the bigger diameter than those of the cell walls and allows the passage of large molecules such as proteins, nucleic acid, etc. The nucleons with the nucleus contain ribonucleic acids RNA in granular form. Then chromatin, the regular clamps and three thread-like material distributed through the nucleus is the most important component of the nucleus. This consists of DNA. The chromatin immediately before cell division organizes into the simple small thread-like structures known as chromosomes, which consist of nucleoproteins molecules. Most of the DNA present in the nucleus occurs exclusively in the chromosomes. Then third, mitochondria. Mitochondria are the largest compound in the cytoplasm. They contain about 35% of the total proteins of the liver. In addition to proteins, they also contain lipids 25%, most of which about two-thirds are phospholipids and a small amount of nucleic acids. They are the poorly house of the cell and each cell may contain from 50 to 2500 mitochondria depending on the respiratory activity of the cell. Now mitochondria have two membranes. The outer membrane is smooth while the inner membrane is folded to form ridge or crestate which extend into the matrix of the mitochondria. Two spaces visualizing interstitial space and the matrix space are therefore developed. Then we can discuss the mitochondria contain a large number of enzyme systems known as cyclophores which are involved. First, oxidation of pyruvic acid, acetyl coenzyme A through Krebs cycle, fatty acids, amino acids. B. Electron transport and oxidative phosphorylation. And C. The synthesis of fatty acids. They also contain enzymes concerned with the biosynthesis of amino acids, porphyrins, phospholipids, HAMs, and sulfuric acids and urea. Then fourth, endoplasmic reticulum. Electron microscope have revealed an extensive membrane system in the cytoplasm called endoplasmic reticulum. This membrane is constituted of protein lipids double layer and is very developed in tissues with active protein synthesis. It was first reported by Porteo in 1945. Then Golgi bodies serve as means of producing and maintaining the internal membrane. The interior of this membrane is concerned with the perinuclear space and sometimes also with extracellular space. So, this exchange of material occurs the process of diffusion, active transport and pinocytosis. This and the surrounding matrix are not only means for specific metabolic activity but also provide the means of communication between the nucleus, mitochondria and ribosomes. The important function of this membrane and cytoplasm matrix are transport of nutrients and metabolites into and out of the cell. Then regulation of protein synthesis and helps in the transport of proteins. Then glycolysis. glycogenesis, pentose phosphate pathway, fatty acid synthesis, and lipid synthesis. Then we discuss the ribosomes. Clute 1943 came across with some basophilic granules rich in the nucleus acids ribosomes, and Brown 1953 could observe the granulars in the root cells of beans and the electron microscope. Pellet 1905 isolated these granules and found them to be rich in RNA. He was awarded Nobel Prize in 1974 for this work. So these granules, being rich in ribosome nucleic acids (RNA), came to be known as ribosomes. They occur outside of the membrane and endoplasmic reticulum. The size of the ribosomes range from 50 to 20 millimicrons, means 10 to the power minus 6 millimeter, and the diameter varies from 150 to 250 angstrom. Ribosomes are composed of RNA protein complex. They also contain lipids, mainly phospholipids. They occur separately or in a polysomal cluster. They consist mainly Perkins of the two sides, one with sedimentation constant of 50 S. So, ribosomes also process steroids reductase responsible for metabolism of cholesterol and steroids hormones, phosphates, hydrolase and hydroxylase, glucoanyl transferase and ATPs, etc. Then centrioles. Centrioles are two short cylindrical structures known to exist to either side of the nucleus at right angle to each other. They are not bound by any membrane. They help in the equal division of the chromosomes by taking them part. They are therefore responsible for equal distribution of the characters in the offsprings. Lysosomes, as shown in the diagram 4, types and their origin. This is shown in the diagram. So we discuss lysosomes. So Belgian biochemist Christian D. Dewey in 1955 discovered lysosomes, awarded Nobel Prize in 1974, and he was the first to use the term lysosomes. Lyso means digestive, soma means body. So lysosomes are rich in many hydrolysis enzymes. The size mainly means diameter 0.4 micron of the lysosomes is in between that of microsomes and mitochondria. They are surrounded by lipoprotein membrane. So lysosomes contain digestive enzymes such as peptine, acid phosphate, ribonucleics, deoxyribonuclease, catalase, collagens, alpha glucoside and beta galactosides, alpha monosaccharides, phosphoproteins, phosphates, etc. Unless the important membrane of the lysosomes is ruptured, the enzymes are not released into the cytoplasm. Hydrolase in lysosomes are separated from their substrate by means of lipoproteins membrane, and only when this membrane is ruptured, the hydrolase either the cytoplasm and destroy the bacteria of foreign particles when the latter enter in the cells. The rupture of the lipoprotein membrane occurs under conditions of cytolysis, which includes fat solvents, detergent, proteins, lactamase, acid, and pH and high temperature. Then lastly, we discuss the cell part of cytosols. So Nardi introduced the term cytosols of the cell sap. It is the unstructured soluble protein of the cell. It is the supernatant that remains after centrifugation of mitochondria, microsomes, lysosomes, and nuclei. So, cytosols mainly contain the enzyme system involved glycolysis, fatty acid synthesis, pentose phosphate pathway, activation of amino acid in protein synthesis, and catabolism, purines and pyrimidines. So thank you all of you.